Yeah, you're good. Yeah.
Okay, so we're good now?
Yeah, it's recording.
Okay. Okay, so for those who are watching the recording, you missed the introduction, but long story short, I just introduced myself and basically how to learn R, which is a package called Swirl. I'll go back real quick. And here's what it looks like. You just download this package. And this is basically how I learned R and how I recommend everyone to learn R. And anyways, so let's continue. So as I was saying, there's many different types of RNA sequencing, but it all depends on the lab that you're joining. And for my lab specifically, we use single-cell RNA sequencing. We're going to get into what single-cell is and why it's so big nowadays. But I would just recommend you guys to just explore all of these and just figure out like, okay, this is something that I like. This is something that I don't like. Something that single-cell does really well in is that the starting material is super easy to use. If you look at the columns that says starting material, all you need are the cells. So literally what we do is we just freeze our tissue, and then after it's frozen for a set amount of time, we put it into the machine, in a series of machines, and then it does the rest of the work for us. It gets all the data for us. Like, that's so easy to use. And that's also why it's expensive, because you don't do that much manual labor. And so that's where the expense is going. So yeah, let's just go ahead and move on. Hello. So why is single-cell RNA sequencing so big nowadays? I wanted to give you guys this example. So we have an aging population, and every year our life expectancy gets older and older. And so as we get older, there will be more diseases that we have to take care of. And it's getting harder and harder to understand these diseases because they get more and more complex. And so a prime example that's really big in the industry right now is Alzheimer's disease. And that's actually what I study as well. So if you have a healthy individual who's 90 years old, and you have an individual who's 91 years old with Alzheimer's, how are you supposed to like ethically, compare their brains? So that's where single cell comes in. It looks at the individual cellular level between two individuals, and it looks at the makeup between them and compares them. And so if we take a look here, we can see like this is just a very broad, this isn't every case, but for example, with somebody with Alzheimer's, we might see an increase in microglia, a decrease in astrocytes, a decrease in oligodendrocytes. And this was made possible because we can look at the cellular populations of the individual with Alzheimer's versus the person with a normal brain. And so the question is, you can ask questions like, how does Alzheimer's play a role in cellular populations? Or how does Parkinson's play a role, or any other neurodegenerative thing? You don't even have to do brains. You can do lungs, you can do hearts. And I know the field is really big in blood research as well for single-cell RNA sequencing. So it really just depends on what your focus is and what you want to study. Are there any questions so far on why we do single cell? Okay, so yeah, we're just looking at the individual cellular makeup of an individual, and we're comparing it to other individuals. So just a little bit of background. So now that we know why we do it, well, what is single-cell RNA sequencing? So it was first developed in 2009 by Fuqiu Tang, and I don't even know how to say his name, but Fuqiu Tang at the University of Cambridge. And it wasn't really that big at the time because it was really, there was a lot of criticism. It was very skeptical. And then about 5 years later in 2014 is when it first got commercialized and it was being sold to bioinformatic labs. And so one thing that makes it so amazing is you can study many types of genes at once across many cell types. Other RNA sequencing doesn't have that power. Like, there is some similarities, but you can't inspect the individual cell types. So another popular sequencing is called bulk RNA sequencing, which many of you have heard of and maybe have worked with some of the data. But the reason why bulk RNA sequencing is it's still great, but it's not as good as single cell is because bulk RNA sequencing takes the average expression of the genome and displays that as the data, whereas single cell doesn't take the average. It looks at the individual cells, and those are your data points. So that gives you much more power when you're making conclusions. Like, you can make more powerful conclusions at the end of your research when you can look at individual cell types rather than the average expression. So that's really what single cell is. It's looking at the individual cells, and I've said that over and over again. And so SURAT is a pipeline that was produced in 2017. It was made by Satija something, I don't know his full name, but his first name is Satija. And basically he made the SURAT pipeline, and this pipeline basically boomed the industry for single cell. It's not in just RStudio, it's in Python and many other languages as well, but it's mainly used in R because scientists just use R. It's kind of like the science program, but Python is very popular as well. And this is what we're going to go through today. We're going to go through his pipeline, but there's so many other types of single cell sequencing. So if you disagree with some of the metrics or some of the methods, like feel free to explore other pipelines. But this is what my lab uses and what many other labs use. They use the SURAT pipeline. And I made this graphic on the right, and I'm very proud of this graphic. It took me a while to make, but basically what we're going to cover shortly after this is we're going to cover phase 0. So this is all the wet lab stuff, like how do we do single cell? And there's many different methods to this. Like there's no one set way to do it, but I'm going to just walk you guys through like a basic fundamental step-by-step. And then if you find other methods, like sure, you can do those too. And then we're going to go through, we're going to talk about it later, but like there's other phases too, to like the data analysis portion. So we're going to go through those later in the hands-on section. But for now, we're just going to cover like what happens in the wet lab stuff. So are there any questions so far about like what single cell is? Like I'm trying to make it as clear as possible that like that so that we all understand. But if there's no questions, then yeah, okay, so let's just move on. Let me check the Zoom chat, one sec. Okay, sweet. Okay, so one of the biggest companies for single cell work is 10x Genomics. And the reason why is they basically just have a monopoly on the industry. And so they produce most of the data. They produce machines that scientists will pay for. They're very expensive. And the reason why I chose them today is because you'll probably be working with some of their data in the future. And it's important to know like how they even get the data. And so I'm going to just walk you guys through this step by step. And I have it written down here because I don't want to mispronounce anything. And so first off, you start with 10x droplet-based beads. And these beads are basically used, they're in the top. Hold on, let me get my laser pointer actually. Is there a way to do pointer options, laser? Here we go. So you'll start with these beads, and these beads are important because it lets you identify cells when you tag cells with these beads. You're basically going to attach your cells with these node beads, and then that way you can say, okay, this is cell 1, this is cell 2, so on and so forth. So we call those 10x beads. And we're going to basically insert them with our cells in a series of microfluidics, and that's going to separate out all of the cells and beads together, and it's going to put them together. So you can see it right here, like how the cells will attach to these beads. So here are the cells, and they attach to the beads, and then they move into these things called wells. And for each well, we have one cell and one bead. And that's how we separate it and get it to the single cell, like how we separate them each from each other. And we submerge them in oil. So oil will make sure that they stay separated and that they don't recombine. Because if you put them in water, they will recombine. And so that's why we have to put them all in oil. And so these are the first 2 steps, and they don't really take that long, but usually the machines will do it for you. Like all you have to insert is just your cell tissue. And so then after we insert all of that, let me scroll down here, one sec. So the result that you'll end up with is you'll end up with these wells, and they'll each have these beads on them, and these beads will make up a library. And you'll have your library of beads and cells, and you'll have something called a UMI. So UMI just stands for unique molecular identifier, which is unique to each cell type. So now that we have each cell labeled 1, 2, 3, 4, 5, and we have each cell labeled with its UMI, we can create a database called a count matrix. We're going to make a database that has all of that information together. And so that's what this graphic is displaying here. Like it's showing the cells right here, and we have our artificial 10x code, and then we have the unique cell identifier. And so with all of this is where we'll really begin to start our data analysis and preparation. But all of this is called the library. So when somebody refers to like, how big is your library, or how big is like how was your library prep? Like, this is what they're talking about. They want to know, like, was it clean? Was there any issues? So when they're like, yeah, my library prep was good, like, that's what they're referring to. They're referring to this process, like, was there any issues? This is basically just called library prep, which I'm going to display in the next area. Yeah, what's up? Yeah, okay.
So, for the barcodes, is there a specific oh, you mentioned there's a specific barcode for a specific cell, but how do the cells recognize these?
Yeah, that's a really good question. If for those online who didn't hear, she's basically asking, how did the cells recognize each barcode, right? Am I getting that correctly? Yeah, I don't know how the cells recognize those, but I know that for the most part, all the human, like all we want to recognize are the 10x barcodes, and these unique molecular identifiers aren't that important. We just want to know which cell it came from, and we're tagging it with 1 to keep track of it. 1, 2, 3, 4, 5. I don't know how the cells, it might have to do something with like some methylation thing, but I know there's some poly-A tails that are also used in some recognition, but I don't know exactly how those work in.
What if there's 2 different types of cells?
Yeah, so you're saying they might have the unique or same UMI or same 10x speed, because it's impossible to have the same 10x speed because it's only 1 per, well, it's not impossible, but nothing is impossible in biology, but those are called doublets. So when something is doubled in this, you have to use a function, which we're going to get into later, but it basically separates out all the doublets and it removes them from your data. So that's what's called quality control. When you go in and you look, how many doublets do I have? And let's just remove them. But that was a good question. I mean, sometimes you'll run into problems where you'll have 2 cells in one well, or you'll have some mix up with the barcodes. It doesn't always attach correctly. And then that's when you go in and you do quality control, and then you're going to be throwing away data, but it's also dirty data. So you're not really throwing it away. You're basically cleaning the data. Yeah. And either something happened, like mistakes happen, you know, there's human error. Maybe you just didn't follow the protocol correctly, because all of these have certain timings. Like, it's not like back to back to back. There's steps in between all of these steps too, but this is just a fundamental workflow. Like, you know, you may have to spin it down and then, you know, isolate something. I don't know. I mean, I'm not a wet lab scientist, but from my understanding, this is the workflow for 10x genomics. And there are different workflows as well, but yeah, you might not even be using 10x genomics. You might be using a different workflow later on that does something else, but for the most part, it will look similar to this. But yeah, good question. Okay. So moving on. So we just covered the first 4 steps, and we got a little bit into library prep. So after library prep is really where the nitty-gritty work starts to happen. And we're going to just skip deep sequencing because this is a giant beast in and of itself. But what I wanted to focus on today was data analysis. So data analysis has many steps to it, and I'm going to show you here what I'm talking about. So we just covered phase 0, but the data analysis portion has, okay, just to give you guys a frame of reference, like within each of these, there's like 3 or 4 steps within each of these. So like, for example, when you do quality control, this has about like 3 or 4 steps within that. And then within normalization, like this in and of itself is its own thing as well. So like, if you guys are looking for like a research project or something like to do, you literally could make your PhD off of like, how do I optimize clustering? How do I optimize quality control? Like, all of these things are it's such a new field that, sorry, I'm a little sick. It's such a new field that like you can go in and you can do this stuff and produce it and present it in front of people. And if you have good stuff, like you can make your PhD off of it. It's like, that's how new of a field we're talking here. And so what I am working on right now is I'm working on marker genes, but other people are working on clustering, which is more like they're applying machine learning to it, or they're doing some sort of automation. And so this is the pathway that we'll be talking about today, but we're specifically going to be talking about these things because these things are the most important things that you don't want to mess up when you do your single cell. Because if you mess up like any of your quality control, your downstream analysis will be completely ruined. And if you mess up, you know, normalization, imputation, like any of these things, they're very important. So, and this is stuff that you can't really find online. Like if you Google, like, how do I do quality control? Like, it's very hard to get answers like that. Like, even if you ask ChatGPT, like ChatGPT doesn't know. it'll give you some generic response from like 2 years ago, but it won't stay up to date. And I feel like, you know, there's talking to other scientists about these kind of things, like it can be very helpful. Like, how do you do quality control, or how do you do this? And then you can compare that to other people. So far, I mean, that's all I had for the lecture. And we're going to take a quick break to get some lunch. And then afterwards, we're going to go through a step-by-step. I'm going to show you how to download data, show you how to do everything. And we're going to go step-by-step after we get lunch. But are there any questions? Like, you know, do you guys just have any broad questions at all? Like, are we confused about what single cell is, or why we do it, or anything? Anyone in Zoom?
So what we are doing is there's a biological sample.
Mm-hmm.
We are converting into like what machines or our program can understand.
Yeah.
That's it, broad.
Yeah, so we're taking a sample. It's going to be frozen. I mean, not literally right now, but in the lab, you would have a frozen sample. You put it into a machine. It grinds it down with water, and then you take that out. You spin it down in a centrifuge, and then you take out the tissue. You take out the cells. Sorry. Right now, you just have cells, and then you put it into a machine. Again, like it's all automated. You just are moving it from machine to machine. Eventually, you'll end up with a bunch of cells that are isolated with beads on them that are identifiable by the 10x machine. And then that will make a database for you. It's called, I mean, it has many different names, but SURAT calls it a SURAT object. That's the pipeline. They identify it as a SURAT object. It's just literally just something, it's just a database that has the gene. It has a gene name with the cell, and then the identifiers, like where it came from. That's all it is. And then we're going to be taking that, and we're going to make like a map out of it and show like, okay, this cell is really similar to this cell. This cell is not similar to this cell. And this can lead to breakthroughs. Like if you find a subtype of microglia that's not different, like if you find a subtype of microglia that's very different than the other microglia, then you can be like, okay, this is strange. Like the genes from this microglia, they're not even close to that microglia. Like what's going on here? And that can lead to discoveries. You can even discover like new cell types. Like, there's honestly way too much information. Like it's honestly very overwhelming, but you know, it's a good start. I think give it like 5-10 years. This is probably going to be mainstream, and it's already becoming mainstream, but not many people know how to do it accurately. And I'm not saying I know how to do it accurately, but I've done a lot of research in it. So I hope that I can translate some of that, but yeah. Yeah. Mm-hmm. Yeah, not just transcriptomics. Like you can do spatial single cell, or you can do all types of single cell, but what I specialize in is transcriptomics. So yeah, let's go ahead and take a break. We'll do like a 10-15 minute break, or how long do you think? I also left up a joke here. So if anyone can figure it out, you get a bonus point. As Dr. Wang says, you get a bonus point, and then doesn't give us the bonus point. But yeah, I'm gonna grab some food. Anyways, for those on Zoom, just let me know if you have any questions, and we'll be back in. Let me see what that is. Yeah. Do you want to do that? We'll be back at 1:42. Yeah, whatever. Okay. Data set integration first of the samples, or integrate after? You know, okay, and now you're getting to a really good question, because integration is very controversial within single cell. There's some people who integrate at the very start. There's some people who quality control and then integrate. And it would be incorrect of me to tell you that there's one way to do it, because at this point there is no correct way, because there's two sides to the coin. Both of my mentors, this is a perfect example. I have one mentor who's a postdoc, and he likes to integrate all at the very start. And then I have my PhD mentor, and he likes to integrate after quality control is done. And they have their own disagreements with each other about it, right? And so there's no correct way. What I personally do, though, is that I integrate after, no, all at the start. So you merge all of your data, and then you do quality control. For me, the reason why I do that is I think it's just easier. but there have been studies that show that integration before can lead to like extra dirty data, which I don't know. I mean, I don't know. Me personally, I don't have an answer to that, but that's just what I do right now. I know there are some people working on that specifically, and look, you can even make that your project. Like if you wanted to get, you know, if you want to put together a project and show the differences of integration, like that's a perfect example of what I'm talking about. Like everything in this pipeline is so brand new that like you can make that a project. Yeah, hopefully that helps answer your question, but there's no real way for integration right now. I mean, there's two sides to the coin, and yeah. But anyways, let's resume. So let me pull up my. Okay, so I'm gonna start us out with this presentation, but then I'm gonna move to RStudio afterwards. So let's go ahead and scroll down here. Okay, so oh yeah, let's talk. I didn't even see this. So the answer to the joke was that because I had too many general issues and needed to express. Okay, that was cringe. That was super cringe. I'm sorry. You want to take a look again? Why did the single cell RNA sequencing machine go to therapy? Because I had too many general issues. Need to. I'm sorry. You know what? I'm not gonna lie. I just Googled that one. Okay. So let's. All right, Zahidi, I'm gonna need your expertise on this. So how do I? Why is it not presenting? Resume slideshow.
You might want Richie.
All right, Sujay, get in here. So how do we resume this? It looks like it's already resumed on the screen.
Yeah.
Is it on Zoom?
Yeah.
Oh, okay, we're good. We're chilling. We're chilling. All right, whatever.
This is on the Q&A slide.
Oh, you know what? You're right.
I think it's the wait. I think
Okay,
you change your
click it up.
windows here.
Yeah.
Oops. We can stop sharing for a second and then.
Yeah, this is some crazy stuff, bro.
Wait.
Zoom is not my favorite.
Oh, okay.
Is it working?
Wait. No. Here's your presentation.
It's fine. Take your time. I mean, you're doing a better job than what I could do. Nope. Oh, there we go. Nice. See, does it look okay? Nice. There you go. Okay.
Yep.
So I have a long presentation, but instead my colleagues here in the club told me it's better to go through this manually. So I'm gonna just show you guys how to download this data first. So basically, you're just gonna go to this website. Hold on. Laser pointer. So you can go to this website, 10xgenomics.com, and I'm gonna show you guys on my screen too later. But you're eventually gonna go to you're gonna go to something like this, and this is outdated as well because they recently just changed their website. So I'm gonna manually walk through it after. But we're gonna go here and we're gonna download something called Cell Matrix HD F5. The reason why we're doing this one is because it's a smaller data frame, so running it won't be too long. But you can try any of these other data sets if you wanted. Like, let's say you're on the supercomputer or something and you want to try it, like feel free to do your own analysis. But for the purpose of today, we're just going to do this one. And just for a little background, this is a nuclei isolation, and it's looking at the brain of mice. So without further ado, I'm going to show you guys real quick how to download this. Oh yeah, thanks for the link, Sahidi. Let me change my screen real quick so that you guys can see this. So we'll go here, share. Can we share? Unshare and then, okay. So stop share. 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 Can you see it on Zoom? Not on there. Okay, so Zoom can see it, right? Okay, well, I'll go ahead and show Zoom, and then if anyone in person needs help, I can, or do you know how to fix it? Okay, it's showing on Zoom, but it's not showing on the PowerPoint up there. What do you think? Isn't that so weird?
I think it's the. Okay, well,
Yeah, yeah. No signal.
okay, now you open the Chrome tab on this one, maybe. Yeah, people can see your screen.
We're good?
I think we.
Okay, we're good. So we're gonna go to 10x Genomics, and then you're gonna go to the data sets, and you're gonna scroll down, and you're gonna click on, I think, cells or nuclei here. Hold on, let me take a look. Cell nuclei, yeah, cells or nuclei. Let's sort by nuclei. And then we're gonna go and look for, here it is, 5K right here. So it's gonna say 5K adult mouse brain nuclei. Make sure you guys can click that. So we're gonna click on this one, and then, so it's gonna ask you to make, it's gonna ask you to make a thing. Of course we're going to, because we're down bad for the data. So let me just go ahead and, I already have an account, so I don't know why I'm doing this. Essentially, you're gonna make an account real quick, and then you're going to download the file that I showed on my slides. It's gonna be called Feature Cell Matrix HDF5 filtered. It's gonna give you a tar file. So once you download this tar file, you can go to a website that I like using called where? How do I move this? I'm sorry. I mean, I don't use this much. Huh?
Yeah.
More hide.
No. My bad. This one.
There we go. So once you download your tar file, let me know if you have any questions, but no, it should just say, where did it go? Yeah. Yes, you can sort by nuclei here, and then you can scroll down. It's going to be version 3.1, adult mouse brain nuclei. You click on this. You're going to have to make an account. I'm not going to make one, but what it's going to look like is it's going to look something like this on the right. It's going to have a bunch of different files, Sahidi, and you're going to download the one that says Feature Cell Matrix HDF5. So once you download that, it's going to give you a tar file. And did it give you a tar file? So what you can do to just quickly, H5. Okay, so then you can try, okay, so try the cell matrix filtered. I'm sorry, I might have highlighted the wrong one. Can you try that one, Sadie? This bottom one? So that's the tar file. Okay, I'm sorry, guys. So this one here, let me move this down. So feature cell matrix filtered. This will give you a tar file. And then we go to easyzip.com, and I'm too lazy. You can do this all in R code, but I'm way too lazy. I just do it the faster way. Just you go to any zipping website and it will completely unzip it for you. So let me pull this up. And this is the boring part about doing experiments is getting all your data. I don't know why it's not working. Easy zip. Oh, I misspelled it. Okay. Yeah, easy zip. Here we go. So then you select your tar file. So I don't have it on me, but you're gonna just go and I might have it on me. No, I don't have it on me. But you select your tar file and then you download the folder that it gives you. And then that's it. You're done with the data downloading. So are there any questions so far? Like, let's get this sorted because I want to make sure whoever wants to follow along, I want to make sure that you can follow. So if you have any questions, just let me know and I can come take a look on Zoom. Oh, am I still connected on Zoom? Where?
It was good.
So how do I open the Zoom chat? Is that?
Oh, you could escape.
Have you used Zoom before? Yeah. You're so smart, dude. Thank you very much. Okay, so I'll give it like 2-3 minutes and then we'll continue. But yeah, make sure you just have a folder and within this folder, it should have 3 things. It should have like barcodes, features, and something else. It should have like 3 files within that folder. If you have those, you're good to go. No. You should have a folder that has 3 tar file or 3 zip files in it. And this folder should be like filtered, feature, matrix, or what is it called, CD? Yeah, but what's the folder called? Yes, that's what you should have. Yes. And within that folder, you should have your 3 zips.
Yeah.
Here, let me come take a look. If you guys have any questions, drop me a message so that we can get it sorted and then we'll begin our analysis. Let me go help them real quick.
Yeah. There's a specific command you use to get a tar dot. So it's RXF.
Yes, yeah.
Yes, you can just sort of scroll.
On Windows, but I don't have the
minus
program on my
Windows.
So I
go here.
So on Windows, it comes with that one zip. Actually, at this point,
We
I
just
just
do
because I have like a
dash X.
version of Windows, so like it doesn't come with all the basics. So I have to get workarounds like this. Seven zip, yeah, that's what I have in my home PC.
Oh no, it
Is
says
that one too?
5K. Oh, it's an extra A in part.
Sorry.
Oh, it's an extra A.
It looks like everyone online is good, so we'll just wait on those 2 and then we'll begin. Open my R Studio. They're gonna have it downloaded here. There we go. So I'm gonna go ahead. This is my GitHub. So to download the R code, you can just go to my GitHub here. It's just, let me see this. Can you see it, Sadie, on Zoom? Okay. You cannot. Okay, one sec. It's on a different window. One sec. One sec. I got it. So here's the link to my GitHub, and I'll drop it in the chat as well. But basically, you can just go here and download this brain.r. And this is just the R file to run everything. So I'm going to go ahead and download it here. So you just click on Brain, and then you can just go here and click download. So let's open this. You can't see this, right? Okay. So let me change the screen again. 1 sec. Stop share. Sorry, guys, I'm trying my best here. Let me do screen. I'm just going to share the screen. Yes. Took extra precaution. Okay, so here it is.
I would just.
How's it going over there?
Oh, yeah.
Sweet. Swaraj, did you get the R file? Okay, all right. Oh, don't look at this. This is my R file. What step are you on? Yeah. So then to go from there, it should say brain.r. In the top right, you hit download.
Right. And so on your links.
Okay, right. One sec, sorry, we'll
I'm
on my links.
And so if you log into HRC.
No, I haven't, I know from my own.
Oh, yeah. Yeah. So you can open that data. And you can just download it. File.
Let's check the chat. Nope. Sarah. Okay, thank you. Are we good over here? We're all good. Okay, now let's begin. So, oh, see, look, I've got to install some packages. So first and foremost, we need to make sure we have these packages installed. Can we all see on Zoom as well? We can see this. So install.packages, make sure you guys have these as well. Sorry, there needs to be an S here, but I think I have Dippler as well, but let's do patchwork. This should work. Yeah. That's fine. I need to install Surat, I guess. I haven't ran it on my laptop. So this would be a first, but we'll do it together. Install. You can do it all in one line, but I'm just lazy. So Surat is the pipeline we'll be using today, but there are many other pipelines that you can use. I just know Surat is one of the most popular ones, so definitely want to get some experience with that if you want to get into single cell. And I tried to annotate the code as much as I can. So you can just follow most of the annotations, but oh yeah, these are completely out of date. I've got to update this, but whatever, it's fine. Okay, so let's see, did this install? It looks like it installed. Okay, perfect. Let's do Surat. And my laptop is very outdated as well, so things might run a little slower on this guy, but it's fine. Okay. Should be done soon. Okay, it's not done soon. I'm sorry. That was a lie. Yeah, for sure. And also I know that my laptop is probably the slowest laptop in here, so yeah, just bear with me. Yeah. Yeah, we don't need to update R, just hit no on that. Yeah, yeah. Sometimes I'll hit do it now and it'll take me to the webpage and I'm like, really, dude? Why would you do that? Wow, this is a little depressing. Failed to look for directory. Hold on, I might have messed up one of the. Hold on, let me check my current working directory. Oh, sorry. Don't think I have Surat in here. That is strange. I don't know why I'm not allowed to. Let me read this error message real quick. Oh, you know what? Package. I have no idea what I've never seen this error message. Regardless, I'm gonna just walk through the code. I mean, it's not important. I mean, you guys can run it on yours and I can help I'm gonna troubleshoot this in the meantime, but let's go ahead and just start running our code. So these first 3 lines we'll read in our library. So just go ahead and read these in. I obviously don't have Surat because I don't know. I mean, I don't know why I don't have Surat, but whatever, it's not a big deal. We can scroll down here to directory. This will just check like your working directory. The whole reason you're using this is you want to make sure that you have this file in here, filtered feature BC matrix. Yeah, you need to make sure that that's in your working directory. If you don't have that, there's some workarounds, but you can also change your working directory with this as well. So that's what I did here to make sure that I can read this in. And so you're going to just run this brain data. You'll read it in. So this read 10x is from Surat, and this is what they use to basically read that count matrix that we were talking about earlier. So we're going to go ahead and read in this data. And so here is where we're going to really start investigating our data. And while we run this, I'm going to try to troubleshoot what's going on with my Surat. I don't know why it's not. I mean, I haven't ran it on my laptop in like literally months, but so let's talk about everything in this Surat object. So counts equals brain data. This is just taking in your data and it's going to label it as a mouse. I labeled it as mouse brain data set. That's what MBD means, but you can label it whatever you want. The reason why you label it project is it's a little bit about what Emmanuel was talking about earlier, but when you're integrating multiple projects, you need to label them so that you know which sample they came from. So like, for example, let's say we were doing multiple mouse, right? Or multiple mice, sorry. You would do like mouse brain 1, mouse brain 2, like you would label them separate things so that you could track from which mouse they're coming from. But for this case, we're only using one data set, so like we don't need to label them like that. So I just did mouse brain data set. Min.cells, this is saying that you're only taking data, you're only taking in genes that are expressed in 3 cells or more. Anything that's not within those, you're going to remove. And then min features equals 200. Some people do 300, but for the sake of this, let's just run with 200 just so that we can get more data. But min features, all it simply means is that in each cell, it has to have a minimum of at least 200 genes. And if it doesn't, then let's just scrap it. And whenever you see features in regards to single cell, usually this just is a code word for like genes. So later on, you'll see like this. I actually have a presentation. I can just go with this. So let me present this. So like it'll say brain, an object of class Seurat with 22,738 features. What this means is like you have that many unique genes across 6,931 different cells. And then within one assay it just means it came from one sample. So like if you integrate multiple samples, it'll say 2 assays, 3 assays, or however many assays. And you're merging them all into brain, which is what we did. And yeah, and so this right here, this 2-dimensional reductions, calculated PCA and UMAP, these are just types of linear reductions that we will do later on. It just stands for principal component analysis, and I don't know what UMAP stands for, but it's just a type of graphing that we'll look into later. Let me actually just go ahead and share this. So let's do this, share. Yeah, I'm gonna swap it. Yo, Sujay, get up here. How do I swap it back to the, because apparently my Seurat's not downloading. How do I swap the presentation around so that they cannot see my notes? You get me? So that they can see the presentation view. Try that one. Because I have it all in a presentation anyways, but like they can just follow along. You get me? Yeah. Yeah, so just skip ahead a bunch. That's good, thank you. Okay, so that was our first step. We're gonna read in all of our data. And then our next step is going to be looking into quality control. And so, Emmanuel, this might help you a little bit, but we talked a little bit earlier about doublets. And this is a big problem within single cell, is that sometimes your cells will just explode because they're very fragile. And so what will happen is you can look at this column that we're gonna artificially make. We're gonna call it the percent mitochondria column. And we're gonna do that with this line of code here, brain bracket percent MT, which we're making a data frame within our data set. And we're gonna basically take all of the genes that have mitochondria dash in it. So one thing to note is that if you're working with mouse data, it's going to be underscore, or it's going to be undercase, because human data is all uppercase. So we're working with a mouse data set, so we need to make our gene name lowercase. But let's say one day you're working with human data, you need to adjust your code so that you know that if this is human data, then I need to change stuff in my code. So this would be one thing that you change. So the pattern, if it was human data, you would make it all uppercase. Anyways, so we're gonna make it lowercase. And then we're going to do a violin plot. And we're going to show each of these features side by side. So basically we're gonna show the number of features. So these are the total number of, okay, yes, these are the, sorry, one sec. The amount of expressed genes within each individual cell, right? And then the middle column is the amount of libraries within each cell. So like the total amount of genes within each cell. And then percent mitochondria on the right is what we just made. The percentage of those genes, which are mitochondrial genes. And we can see that there's a clear double, there's a group that's doubled. Yeah, I can use a laser, one sec. So it's this group that we want to clean first, because we know that, hey, this group is not correct. Like we have a weird abnormality here. We need to get rid of this. So we can adjust our code with this. And Satija recommends you do less than 5%, but sometimes you have to consider like you're throwing out a lot of data. So you can start with 15% and then work your way down. So that's what I did. And that's what one of my mentors recommends we do as well. But some people do 10, some people do 5. But if you're going any lower than 5, then you're throwing out a lot of data. So I would recommend start with 15 or 10 and then work your way there to see how it's going to look. So we're going to run this, and then we're going to keep everything else the same. So we're just cutting off all these other things. Like there's a few other abnormalities up here and maybe some up here, right? So we're going to just cut all those off here with these following guidelines. And we're going to replot them and take a look at our difference. And we can see here that we cut off most of that data, and we cut off the data up at the top that was looking a little weird, you know. And so this is the first step to quality control. Like we're just making sure that there's nothing weird going on here. And you can see down here, like this all came from mouse brain data set. And this is going back to integration. If you compare multiple samples together, you can see like my first mouse was clean, but my second mouse wasn't. But in this case, it doesn't matter because we're only using the same data. But in some cases, you might need to look into why was my first mouse more dirty than the other one. So next, after we do our quality control, we're going to move into something called identification of highly variable features. The reason why we want to do this is if we're looking for certain genes, we want to quickly tell if they're in a certain data set or not. So like a common one for microglia is INPP5D. And INPP5D is really big right now in Alzheimer's because it's, okay, it's back on. INPP5D is very big in microglia. It's one of the activators for like irritation and whatnot. So for example, you can search up certain genes within your data set. Like if you want to see if it's highly variable, you know, this is really important. You can quickly identify what is variable at the top. You just want to keep track of stuff that's variable because the whole point of single cell RNA is we want to see what's changing. So if something is highly variable, it means that in some cells it will be expressed, in other cells it won't. All the stuff down here means that it's generally expressed at the same level throughout your entire data set, which those won't be too interesting because they're going to be similarly expressed throughout each cell type. But these are what's going to be interesting for us. Like, you know, maybe we can track these and see they're all part of the same group. It looks like, I mean, HBA, HBB. So we would have to look into those later and maybe check those out. So the next step is we need to normalize our data. Now that we've identified some genes, we want to normalize. And what I did was, you don't actually have to run this part here, down here, this violin plot, but I wanted to show you guys what normalization does and why we do it. So you can just look at the y-axis here, like the expression for, what is it, HBB-BS, right? The expression here is so variable, like in a majority of the cells, it looks like there's zero expression. But then you have like maybe 30 or 40 cells where it's just the expression is so high. So the reason why we need to normalize that is because when you're dealing with big value like changes like this, your data is going to look really weird if you don't normalize it. So you run your normalization, and the reason why we use scale factor is we're going to scale it by a factor of 10,000. This is what Satija also recommends as well. And this could be something that you look into for one of your projects, but for the most part, the field always does scale factor of 10,000. I don't think there's not really any reasoning why other than it just makes the data more pretty. But maybe that could be something that you look into. And yeah, so you can see here, like now that we've normalized it, the data is much more comprehensible. And yeah, we're going to move on from normalization. And there's many different types of normalization methods too, but we just use log normalize. But there are like 2 or 3 other types that you can use, especially different pipelines. They might use different methods, but yeah. So this is called linear dimension reduction. We're basically going to, now that we've normalized our data, we're going to reduce it further so that we can compare each gene with other genes. And the reason why we want to do this is for 3 big things, right? The bigger your data set, keep in mind that we're using a very small data set. The bigger your data set is, the longer everything will take. So we need to keep things clean, and we're going to reduce it as much as we can so that things can run quickly and it will be cheap to run. So again, this is linear dimension reduction, and all that DIMS means is I just wanted to plot the first dimension and the second dimension. So this stands for principal component 1, principal component 2. And how a principal component works is with each principal component that you start with. So principal component 1 will be very important with the data set. This is going to heavily influence your data set with these genes. Principal component 2 is going to heavily influence your data set, but it's going to be less than principal component 1, and so on and so forth. So principal 3 will still be important, but it won't be as important as 1. And that's how I like to think of principal components. Like the concept is that the further you get away, the less important it is. So you can start cutting off data with that concept. And so what you just want to really look for is you just want to look for linear trends like this. Like you can see like, you know, these genes here are linearly impacting the data, which is what we want. But as soon as you start seeing like curved data, like, I mean, this isn't that bad too, because this is still pretty linear, but once you start seeing PCs that are not so linear, that's when you can start telling like, okay, this PC is losing a little bit of value, so we can maybe think about cutting this PC off. And that's kind of just the concept behind linear reduction. So you want to view all of your principal components, which is what we're about to do with the next code. So I should have it in my R code. And this one might take a little bit to run. It's called elbow plot. But basically what we're going to do is we're going to use something called elbow plot. And this determines where we should cut off our data. So nobody really shows this in their code, like in their papers, which is really weird because I feel like this is very important. But how you want to read this chart is basically what I said is like the first principal component has a lot of variability, right? Look at the standard deviation. It's at 10. So what this means is the genes here are going to be highly variable and very interesting to us. But as we go further and further down, they're going to be less variable, and these genes are going to be less impactful on like the interesting side of biology. So how we read this is you can cut it off, like there's no one set way to determine where to cut it off, but computational biologists kind of agreed, like as soon as you start getting level, like slope of 0, then that's when you want to basically cut your PC off. So that's why I cut mine off at around 25. There are some people who would just do like 30, 40. There's some people who do 15 or 10. Like Satij's the type of guy to do 10 because he only wants to make sure that there's only highly variable genes. But some people don't want to waste all that data, and so they cut it off at 25 or they'll cut it off at 30. But it really just comes down to like what you're willing to throw out and what you don't want to. But for what we're going to do, we're just going to cut off at 25 because whatever. It's just like a demonstration project. So yeah, and like I said, these are all each individual principal components. So each one gets less and less meaningful because it's less variable. And so yeah, I'm just showing here that we're going to be cutting off at this point too. So, yeah, are there any questions so far? Or... Oh, the code is still running. Oh, I see. I didn't include everything in the presentation because there's there's more stuff in the code. Like I really don't know why the startup's not downloading it. I'm not understanding, but I mean, if you guys have any questions, feel free to ask. Like, how do we use this graph? I mean, I didn't include everything in the presentation, but I'm seeing on your screen right there, you have some heat maps. So do you want me to cover the heat maps real quick on how we read those? Yeah. So the heat maps are also used in the same sense. For those who don't know, the heat map is just like it's just a chart that shows upregulation and downregulation of genes. And essentially what you want to look for is you want to make sure that there's good separation between the upregulated and the downregulated genes. So as soon as you don't see that separation, that tells you that the principal component is not worth keeping. So let me actually pull up an example for you guys. Let me go ahead and get out of this. So stop sharing. So let me share my screen again. So I'm going to just show you guys an example real quick. Just something that I find on Google. Hopefully I can find something. So heat map, up and down regulation. Okay. Let's see if there's anything here. Yeah, that's not what I wanted. Hold on. It's in the Cell RNA-seq heat map. Can you actually tell me the code for that? What is it? Dim plot? What is it, Sahidi? Dim heat map? Yeah, that should do it. Okay, here we go. Yeah, that's what I wanted. Okay, here we go. This is what I wanted. Yes. So okay, so first off, does anyone in here know how to read this? No? Okay, so let's talk about this. So there's two things that you really want to look for. You want to make sure that the colors are separated. So what we can see in this picture here is that the top left is all pink and the bottom left is all yellow. But what's going on in the top right? You know, this is a little weird to us. So this would be something that we want to investigate because, well, it can mean one of two things. We want to investigate it or we want to throw it out. If it's in PC1, we would want to investigate it because this is a very important set of our data because it's principal component 1. But let's say it's in principal component 20 or 30. Then that's when you can start saying, okay, this is just like junk. Let's just throw it out. The reason why is because you don't want to have too much upregulation and downregulation in the same quadrant. That means that the genes are not clearly separated, and this could lead to it could either mean one, that your data is weird, like you didn't clean it properly, or two, there is something interesting going on. And so this is what we're doing inside SOIC. We're pasting all those principal components together. And in my code, I think I wrote an annotation that we want to cut it off. We would want to cut off around principal component 7 or 8, because that's when the separation gets a little messy. And from there on and therefore, it is weird. And so that's when we cut it off. So far, are there any questions on this? I can keep talking about this. Yeah, what's up? Yeah. Okay, so as far as my understanding goes, and I don't know precisely why they have 4 quadrants, but to my understanding, it's one of these are upregulated and one of these are downregulated. And as soon as you see, so like, for example, it breaks, the reason why you have these genes here is it takes the top 10 genes and it takes the bottom 10 genes. So these bottom 10 genes are the least variable in the first component. Let's just say this is the first component. In the first component, we have these bottom 10 genes, and these bottom 10, these are the least variable genes. But these top 10 genes, these are the most variable genes. And I need to go and look one more time to clarify, but essentially one of these will be upregulation and one will be downregulation. And you want to see like, okay, these were all upregulated, and maybe there's some weird separation thing going on here, like they weren't all downregulated. Whereas down here, we have these not so variable genes. These were very downregulated. And I mean, I don't know exactly what's going on here, but that's kind of the way to read this. All you're really looking for is the separation between upregulation and downregulation. And you want to see like, is the separation good? If it's not good, we can probably throw it out. If it's in a high principal component, like 1 or 2, you need to investigate it. And that's why we go all the way to like 8 or 9 within the dim heat map. Let's see if there's any other good ones. Yeah, I mean, some of these are like crazy. Like this is just so, I don't even know how they got this data. That is some crazy data. But yeah, something that would look good. Let me see if there's any good examples. Yeah, so this one's pretty good. So like you can see PC1, PC2, really good separation. PC3, it's getting a little weird, but it's still good data. PC4, like this is when you can probably start considering to cut it off, but by PC, what is this, 6, I would definitely cut off anything past PC6, because at this point it's getting, you know, you can't even tell what genes are being regulated in which direction. And each one is case by case too, like each experiment has its own reasonings for things, so you don't have to cut it off at 6, but me personally, I would. So Sahidi, is the code still running for you? What step are you loading in? I think it's the elbow plot, right? Jack straw. Jack straw is used to calculate the elbow plot, right? Yeah, so is that still running for you? Okay. Right. Okay. Okay. Okay, sweet. So you're done with your elbow plot? Because I don't see it on your. Oh, sweet, you got it. Okay, sweet. So we should be done with the elbow plot. So Keep in mind, we haven't done any analysis yet. This has just all been cleaning so far, like deciding where to cut off our data. So let me go ahead and close this one more time. Sec. Open this back up. And we're almost getting to the annotation side of things. So let me share this. DJ, get up here. So stop sharing, share. PowerPoint, right here.
No. You want to start the slideshow first.
Start the slideshow and then we share, right?
Yes. Do you want the presenter view?
No, it's fine. I don't need that. As long as they can see it. Can we? Oh, no, they can only see the presentation.
Yeah.
Not on Zoom.
Oh yeah, because we're not sharing it yet.
Sweet. I think that worked. Sweet. Thank you. Okay. So now we're going to plot our UMAP. So this is something that you'll see commonly in single cell. They call these UMAPs. All it is, it's literally just taking your genes and it's putting them together and it's clustering them together. It uses something called k-means clustering. So k-means is just a type of clustering. I don't need to get into the specifics of k-means, but there's many types of clusterings and you can even change it in Seurat if you wanted to change it. But most people just use k-means because that's kind of the standard that's been set. But if you have a reason to change, you can. Anyways, we're going to use UMAP and it's going to plot our data like this on the left. Let me go ahead and drop the laser. So here we go. So once you have this data, let's go ahead and break this down. So our y-axis and our x-axis, we have these things called UMAP1, UMAP2. I mean, this is just an arbitrary title for the axis. Like this has no meaning other than it's just telling you it's a UMAP. So you don't have to think about it like x and y axis. All you want to think about is the location within the graph. So we have these things over here. This is our legend. And we have 0 through 24. So does anyone know what the 0 through 24 could mean? Like what are these? Yeah, that's close. These are our clusters. So we have 24 different clusters of cells. And how the numbering works is 0 is your biggest cluster, and 1 is your second biggest cluster, and so on and so forth. So what does that mean? That means 24 is your smallest cluster, and it might not be interesting to you because it's your smallest group of cells. Likewise, 0 might not be interesting to you because it might just be a giant group of cells. Like for example, here's 0. But 0 is so far away from everything else that like it might not be interesting to us. We have to investigate. But this is how you want to read UMAP. You basically want to look at cell types and where they're located, which we're going to get into. That's our very next step. But first, before we get into cell types, let's say you were interested in just genes. Let's say you wanted to look up microglia markers. Okay, this isn't a microglia marker, but let's say you want to. You can type in, instead of like this line here, you can type in just genes and it can search for just genes and where they're located and how they're related to one another. And this is very powerful too. Like let's say you're doing a study on a certain type of gene within your tissue. You can just track that gene and say like, wow, look at how it's related. Like it's not related at all, or it is related at all to other cell types. Like there's a lot of power within this graph. So next up, we're going to mark our UMAP, and I'm pretty sure I included it on my code on how to run this. If anyone's running the code, can you make sure that I included this in there? Otherwise, I can give you the code right now. But I thought I updated it last night so that does it have the numbers on it or no? No, okay.
Oh, I see. So it's just one column. So I don't have to worry about this. Yeah. On right now there should be. We just did 11. Right now we're doing
Brain production label.
Yep, label true. That should have your zeros. Did you run this too?
Yeah, I heard that.
Yeah, we need.
Sorry, does yours look like mine or does
No,
yours?
it looks like hers is just one color and it's labeled.
I think what's going on here is it's labeled all the clusters as one cluster. So this.
Steps are right there.
Let me try something. Can I try it? Yeah, it's probably a step earlier that where we somehow merged. It doesn't even have the cluster in here. That's so strange. Yeah, I think. It's up here. We need to get rid of this. We need to get rid of this. Ident brain. This is what messed it up. Let me try running this. I didn't want you guys to. Your people are too. Hold on. I'm troubleshooting at this point. Provided graph name does not exist. Brain doesn't exist.
Oh, yeah, it was that then.
Yeah, that's the issue.
Yeah.
We shouldn't have run that. I wanted to use it as a tutorial, but we shouldn't have run that. So we need to do it. But now, so what this is doing is I'm trying to rename the cluster, but it looks like it's. So what we could do is just skip that line. So you would have to go back and reread in your data, and then you would scroll down here and skip the idents. I was going to use this as a lesson if you wanted to switch between resolution, but yeah, we can just go past that. So go ahead and rerun it, and then skip the idents brain because it's relabeling everything to and then you can. You don't need to rerun all this, but like you can just reread in brain. Does that make sense? So like you would probably have to go back. Yeah, comment that out. Yeah.
And then we run this.
Oh, there you go. What? So you commented this out and you reran what?
I rerun this fine. And
Yes.
then,
But when I reran it for him, it didn't work.
I don't know, maybe because maybe
Yeah.
something new.
She's right though. How she did it is right. Let me make sure that people.
Well,
Yeah, I think share your screen and show like what lines.
Okay, so for those wondering, there are these two lines. Yes, I have the code. You're right. Let me share my screen. Sorry. Yeah, I was going to use this as a teaching point, but I mean, I guess here it is. So basically, when you're working within single cell, let me change my screen. Before we say anything, hold on. Share screen. Screen. Share. Within. What? Yeah, you're right. CJ, get up here. How do we switch the projector again? So how do we switch it to my computer again? Like, mhm.
Oh, the HDMI.
So I just gotta plug it in again, I think.
Just plug it in and plug it.
Here.
Try that, yeah.
Until you're gonna wrap it up.
10 minutes. Yeah, we're almost done. We're almost done.
Now you can.
Okay, I see. And then I just minimize. I see. Got it. Got it. Okay. Is it still good on Zoom? So this line here, I made an error, and this is my fault. You don't actually need to run this ident_brain, but the point is this metric here, resolution, you can change this to many things. And this right here is a PhD in and of itself. If you go and optimize this resolution, that's a PhD right there. But what I was showing is that you can run it at different resolutions. So like, let's say you do 0.6, 0.7, 0.8. You can switch back and forth with this line of code here, idents right here. And then it will swap back and forth between any resolution. Anyways, let's go ahead and comment these out, okay? Ignore these comments, or sorry, ignore this line. And then you're gonna have to rerun these two, which doesn't take that long at all. Maybe it takes like 20 seconds. And then you should have what I was showing on the chart in my presentation, which is what you have, right? Yeah. So at first, what's the reason behind what? This line here? So the point is, I wanted to show you guys in the tutorial that you can change the resolution. The resolution will literally change your graph. It'll change the amount of clusters you have. The reason why this is important is some people can make their resolution really high or really low. And there's no reason why. They just do it because they want to fabricate their data. The reason why I said this could be your PhD project is because you can optimize this and show there's a set way to do this. You can't pick whatever number you want. You need to pick based off whatever metric. Is that making sense? But for example, the reason, yeah. Yes. Resolution is a parameter. It works like a microscope. Think of it as you're zooming in and zooming out of your data. The higher your resolution, the more you're looking into the cells on an individual level. If you zoom out, yeah,
You increase the resolution, make it more diverse.
Right.
It'll make it less.
Right. And this is, think of it like a microscope. When you zoom in, you're raising your resolution, which means you're going to see more details, which means you're not looking at the big picture, you're looking at specific stuff. So let's say you're looking at just microglia. You want to have a high resolution to look at those set of microglia. If you aren't looking at just microglia, let's say you're looking at every type of cell, you can have a lower resolution to look at everything, right? So that's the point of resolution. This metric basically determines your data analysis, and that's why it's so important. The problem is nobody has set a gold standard on what to use. So this is why I'm saying you need to get comfortable changing between resolutions, because scientists want to see different layouts. You might show someone data and they might not like it. So you just gotta make stuff up. Anyways, don't actually fabricate, but yeah, let's just ignore the idents part. Anyways, we're gonna run these two lines, dim plot, label equals true, and that will label everything. We have 7 minutes to talk about annotation. We're almost done. We didn't get to any data analysis part yet, but this is the final step before data analysis. So this is called annotation. We're gonna find our differentially expressed genes. And how it's gonna work is we run this code called find all markers. What find all markers does is it goes ahead and it finds all of the gene markers for every cell type, and it puts them with each respective cluster. So in cluster 0, you have these markers. In cluster 1, you have these markers. In cluster 2, you have these markers. Gene markers. And what you're gonna do is there's this code that I have here, and this find all markers takes a while to run. So don't worry, but I wanna at least try to show you guys what to do. So you're gonna go an individual, they call this manual annotation. You go and you look at each cluster right here. So cluster 11 on the board. You don't have to do cluster 11, but you're gonna have to do all of them eventually. but let's say you look this up, you go ahead and run this, and the output that you're going to get is going to be a list of markers like this on the right. And these markers, you're going to take these markers and look them up online on any database. So one that I use is called Panglo database. Let me go ahead and show you. And all this is on my slides too, if you want to download. Okay. I just need to hide this real quick. Hide, yeah. So the database that our lab uses is something called Panglo database. So what you're going to do is you're going to take those gene markers, you're going to look them up here, and that is going to tell you the cell type that you have for that cluster. So let's see, what do I have here? I have SATB2. I have these, right? So SATB2, HT here, sorry, H3, HS3. Okay. And then I think this is correct. Now, obviously, you're going to want to put in more than 3 gene markers, but for the sake of the tutorial, I'm just putting in 3. And then you're going to select this to just mouse. And the reason why Panglo database is so good is because it's primarily a brain database. So this is what is going to be great for us since we're using a brain data set. But we can see here that most of these markers were found in osteoblasts and retinal ganglion cells. So we have a few options. I would advise that you label this cluster as osteoblasts. So what that would look like is when we go in here, we're going to label each cluster down here. And we're going to I manually went through before today, I manually went through and annotated everything. And this is what each cluster is representative of. And it goes like this. So you don't have to do it. I did it for you. But this is how I got those cell types. And then what we're going to do is with this code here, we're just names, new clusters. We're just going to rename all those 0s through 24. We're going to just label them with these names right here. And then we're finally going to print it out on our UMAP. And then that is ready for data analysis, the biological analysis. So we did all of that work to end up with this. And let me share my screen again. One sec. Yeah, I'm gonna, one sec. Share PowerPoint. Is that good? So let me go to the very end, I'm sorry. So we did all of this work for this image. So what we can tell from this is where we finally sit down and we try to make biological inferences. So what we can see is that oligodendrocytes are very separated from everything else. So what does that tell us about the genes? Well, that tells us that oligodendrocytes gene markers are very diverse from the rest of the group. What we can tell about this group here is that we seem to have this huge cluster of neurons, but they're very related to these OPC cells. So these are oligodendrocyte precursor cells. And maybe, we can make some inferences about this. We need to look at our data set and what we're studying. But let's say we're looking into an Alzheimer's data set. This would be really important stuff. Like we would see like microglia. Microglia are really correlated to whatever, so-and-so. And you can use this to draw your biological conclusions. And there are many different ways to annotate as well, but this is just how I manually annotate. And also this was just a practice data set, so it's not very realistic, but I just quickly threw this together like a few days ago. And some of the annotations might be a little scuffed, but for the most part, this is how you annotate and how you make inferences. But you're really just looking at the position in the graph. So like astrocytes and macrophages, see how they're isolated from the whole group, but they're kind of related to each other though. And you can even see this little section in here where they're kind of connecting. That could be, right, in and of itself, that could be something that you want to investigate because, they're completely isolated, yet they're so close together. Yes, the laser, I'm sorry. Yep, thank you. Laser pointer. So here are the macrophages and astrocytes, right? So just to reiterate, like they're so far away from everything, but yet they're so close together. This could be something that you want to investigate, maybe in another data set or something where you have more data. You can look at just astrocytes and macrophages. And like another thing too, like fibroblasts, they're completely isolated from everything. T cells completely isolated, but then you have these two giant groups here. You know, this group and this group. So you can draw inferences from this graph. And just to reiterate, like this is a small data set. So some of the conclusions might not be too powerful, but as long as you're understanding the concept behind single cell, like this is kind of what it's all about. You look at this graph after you annotate and you say, okay, like neurons are highly correlated with oligodendrocyte precursor cells. And enterocytes, well, they're connected to this unknown group that it's not even in the database. That's what this group is. Unknown means it wasn't in the database when we annotated. But like, I wonder what this group is about up here, right? Because it looks like they're highly correlated to enterocytes, yet we don't even know what they are. So yeah, long story short, I mean, that's kind of how you do the annotation. Sorry about the project. I mean, I don't know why it wasn't working, but yeah. Huh? Yeah, so the thing about the UMaps is I made this graph like months ago. How it works is it uses like seeds, and when it spawns like a set seed, it changes. Do you know about seeds and programming? How like, okay, so like I made this months ago and I didn't record the seed down, and so it just generated this. I need to in the future for better practice, I need to record my seeds. Yeah. 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 Yeah, I need to, I mean, I made this months ago and I also just threw this up last night. I think don't focus too much on the output. You just want to get the concepts down. Like, what do you do with this graph? The point is what you're talking about. Is this oligodendrocyte population close to other cell types? Is it not? Like, these are things you want to be thinking about. And then you can draw your conclusions from that. Like, oligodendrocytes were really weird. We should probably investigate those. But that was a really good point that you made, and I do need to look into that further. But yeah, I also just wanted to give, before we wrap it up, because we're getting close to that time, but I just wanted to give acknowledgement to my lab mates, because they really taught me like pretty much everything I know. And if you guys wanted to follow me on LinkedIn, I would definitely connect with any of you. If you have any questions on single cell, please let me know. Like, I always like talking about it. I'm not a super duper expert myself, but I try my best, and I would like to learn from you guys as well. So if you want to connect there or reach out, yeah. But other than that, I mean, that's all I had. So sorry about the scuff presentation, but thank you guys. Yeah, thanks. Is there anyone online that has any questions? Otherwise, I'm going to go ahead and. Oh, yes, Sujay. We actually have one announcement. Sujay, get up here. Yeah. Let me. Oh.